looking at the special senses, starting with the sense of smell, then taste, vision, then hearing, and then equilibrium. Both smell and taste are known as chemical senses. In this case, we have different types of receptors that are going to be specific for different types of odorants in the case of smell, or tastants in regards to taste. So in regards to smell, we have 10 million to 100 million receptors for smell or olfaction. They'll be generated within the olfactory epithelium in the superior part of the nasal cavity. So this is going to cover the inferior portion of the cribriform plate back from the ethmoid bone way back when in chapter 6 or 7. It's going to extend along through the superior nasal concha. You can see a sagittal section of the nasal cavity here showing the frontal lobe of the cerebrum and the olfactory tract and bulb just inferior to that, as it lays on top of the cribriform plate of the ethmoid bone where the olfactory nerve receptors will pass through and then basically descend into the olfactory epithelium through the superior nasal concha. So the olfactory receptor cells are first order neurons. They live for roughly about 2 months and then regenerated. They will synapse with the mitral cells in the second order neurons. We have roughly about 10 million olfactory receptors, but only about 400 different types. These are low threshold, fast adapting, unless you have either a new production of odor or a greater concentration of the same type of odorant. In other words, most of you, if you remember walking into, say, your grandparents' house, and you may smell mothballs or apple pie or something that's very particular to that particular residence, you notice after the first couple minutes, you don't smell that particular odorant again. Which means that's a very quick firing off of the receptors, but also very fast adapting. Now on the flip side, of course, if you had a sudden influx of gas, let's say it was released into the house, as it increases in concentration, you would keep still smelling that particular smell. Here's a more detailed version of where the olfactory bulb will have descending axons into the cribriform plate. If we look at the most inferior portion here, you see the odorant molecules down at the inferior portion there. They're going to be taken up by the olfactory cilium. They're going to be extensions of the dendrite of the olfactory receptor cells. Once these odorants will actually bind with the particular receptor, it'll fire off an action potential and travel up through the axons as it synapses with the olfactory bulbs. These are first order neurons synapsing with the second order neurons here. So if you notice, we're not dealing with the thalamus on this one. This is the only special sense that is not involved with any kind of synapse within the thalamus. This goes directly to the olfactory bulb and goes right to the cerebral cortex. Now you have different supporting level cells here too within the olfactory epithelium. These are going to be columnar epithelial cells here, and also have the olfactory glands called Bowman's glands. These will secrete mucus, which helps increase the sensitivity of the reception of the odorant molecules. So the supporting cells are the columnar epithelium. These are more physical support. They hold the receptors in place, basically allow electrical insulation. This basically does the same kind of job as what a Schwann cell would do in the peripheral nervous system. You have basal stem cells that are constantly undergoing mitosis, so you have the quick replenishment of the receptor cells every 2 months or so. The olfactory glands, also known as the Bowman's glands, these are going to help with the transduction of the actual odorant as it binds to the receptors along the cell membranes to allow the conversion of electrical impulses to occur. And again, this also help insulate the area too, especially from any kind of cold or any kind of warm environment. So the pathway for sense of smell, you have the receptors in the nasal mucosa, which will be the first order neurons. These will synapse with the olfactory bulb. This will travel along the olfactory tract and will be interpreted by the primary olfactory area that's found in the cerebral cortex in the temporal lobe. So here you see the pathway here. So again, you have an odor molecule that's going to bind with the receptors. So at the dendrites there, you'll have an action potential of fire. It'll pass through the cribriform plate as the axons will pass in the ascending position, synapse with the olfactory bulb at the second order, and then synapse with the primary olfactory area in the temporal lobe on each side of the brain. So from the olfactory receptor, we have olfactory transduction, which is the binding of the odorant to the olfactory receptor. As I mentioned before, it's going to be low threshold, fast adapting. So unless there's a new smell or higher concentration of the smell, eventually the receptor cell will fastly adapt. Now, usually what ends up happening is once the odorant molecule will bind with the protein, the transmembrane protein, it will activate a G protein, which will in turn turn on adenylate cyclase, which will convert ATP into cyclic AMP, or cyclic adenosine monophosphate, which causes depolarization. This action potential goes to the primary olfactory area in the temporal lobe, also goes to the frontal lobe or the orbitofrontal area for odor identification. So you can see here the odorant molecule will bind with the receptor protein, activates G protein, which in turn activates the adenylate cyclase, which converts ATP into cyclic AMP, opens up the ion channels, in this case sodium, sometimes calcium as well, and that'll cause depolarization of the receptor cell. So next we'll talk about gustation, or otherwise known as the sense of taste. Taste is the other chemical sense. Like scent, basically instead of an odorant, you have tastants. It's going to be a little bit simpler than we have with olfaction, though. There's not going to be as many receptors. Only 5 primary tastes are available. We have sour, sweet, bitter, salt, and umami, which is otherwise known as meaty or savory. Basically, everything else aside from umami are going to be combinations of the other 4 primary tastes. Now, the main difference you'll see with the sensation of smell versus sensation of taste. Smell had about 400 different receptors, each one for one particular type of odorant, whereas taste buds in opposition basically contain receptors for all 5 different styles of taste: sour, sweet, bitter, salt, and umami. We have about 10,000 taste buds on the tongue, the salt palate, the pharynx, and epiglottis, and these will be innervated by cranial nerves 7, 9, and 10 in the young adult. The kids have more, but we'll see in a little bit that kids will lose certain taste buds as they get a little bit older. You'll see 3 different types of epithelial cells found in the taste buds. You have the supporting cells, very similar to what you find in the nasal cavity. You have the gustatory receptor cells, very similar to the olfactory cells, and also the basal stem cells. The basal stem cells also undergo mitosis to replace gustatory cells as they age, or in some people, if you ever burned your tongue before, those receptor cells will die off and the basal stem cells will basically mitopically divide to create new ones. Now, I find different bumps found on the tongue called papillae, which are basically going to be formed by a couple different styles of papillae. Basically, you have valley papillae, which are about 100 to 300 taste buds found all throughout the tongue. You have fungiform papillae, which are going to have about 5 taste buds each. You're also going to find foliate papillae, which is only found in kids. You find these on the lateral sides of the tongue in the trenches. They tend to die off a little bit or degenerate by the time you're around 7, 8 years old or so. Letter C, you see the structure of a taste bud is going to have gustatory microvilli for greater absorption or surface area. Also the gustatory receptor cell. Now this will be a little bit different than how the odorants work. This is mainly used through G proteins and also uses cyclic AMP. Also will detect different types of acidity levels as well. We're dealing with sour and dealing with bitter in particular. They use more so the ion channels more so than using the G proteins found in the sweet, the savory, and the salty. You also see the stratified squamous epithelium to hold everything in place, and there you see the sensory neurons affixed to the basal cell. These little synapses with the 3 different types of cranial nerves we talked about before. So the facial nerve, the vagus nerve, and the glossopharyngeal nerve. So again, we have 3 different types of papillae here, which are the surface bumps you find on the surface of the tongue. You have the valley papillae. There's only about 12 found in the posterior 1/3 of the tongue. You'll find the fungiform papillae, which are pretty much all over the surface of the tongue. And then foliate papillae only found in the lateral trenches on the lateral sides of the tongue. Like I said before, these will degenerate around 7 or 8 years old. And this is why some people will say your taste buds quote-unquote change when you get a little bit older. Basically, as a kid, more of the umami and the sweet taste buds are more prevalent than the other 3, and they kind of balance out by the time you're 7 or 8 years old. You have a different type of papillae found in the tongue known as the filiform papillae. These are a little rougher. These cover the entire surface of the tongue, but these are more so for tactile sense, but there's no taste buds there. So basically that helps you increase friction to move things around your mouth, also help manipulate the food, and also the combination of all the papillae, whether it be filiform or the valley, fungiform, and foliate papillae, all these in conjunction with your saliva, in particular salivary amylase, can start the digestion process and also start to break down the different types of foods, in particular the carbohydrates, so your tongue can actually detect through the taste receptors. So here we see a dorsal area of the tongue showing all the different papillae found. So you have the filiform and the fungiform papillae spread out all throughout the tongue. And remember, the filiform is more so for tactile sense, fungiform more so for taste buds. You've got the foliate papillae found on the lateral trenches of the tongue. These are the ones that degenerate over time, usually by 7 or 8 years old. You have the valley papillae, only about 12 of them found in the posterior 1/3 of the tongue. followed by the lingual tonsil and the palatine tonsils. The palatine tonsils are little pouches on either side that occasionally trap food. A lot of times if they get stuck there, they can form what's called a palatine stone. These become very obnoxious smelling, where bacteria basically forms around the food. It actually can cause halitosis, which is very bad breath. It can be coupled with different types of problems with the tongue there as well. And of course, more on the posterior there's the epiglottis. So as far as the cranial nerves go, cranial nerve number 7 takes care of the anterior 2/3 of the tongue. The glossopharyngeal takes care of the posterior 1/3, and then finally the vagus nerve 10 takes care of the epiglottis, as well as some taste buds scattered throughout the throat. So again, here are the 3 cranial nerves we just talked about. So again, facial nerve will cover the anterior 2/3 of the tongue. Glossopharyngeal number 9 takes care of the posterior 1/3, and vagus nerve takes care of the taste buds on the epiglottis and also in the throat. So we see all 3 of the cranial nerves will take sensory innervation from the actual tongue itself. These will synapse with the different facial nerves and also synapse at the gustatory nucleus found in the medulla oblongata. It will synapse at the thalamus and then also go to the primary gustatory area of the supracortex. One thing of note, if you remember back and looked at the olfactory area, the olfactory area and the gustatory areas are very close to one another. That's why a lot of times we're able to identify or enhance certain types of taste because we're also getting sensory information from the smell receptors at the same time. And those of you who have colds before, which most of you have, or you've had any kind of colds where your nose gets stuffed up and you can't smell certain things, a lot of times your sense of taste is diminished. But both of those areas, both of the primary sensory areas of both the olfactory and gustatory areas work very closely hand in hand. And of course, this also works for recognition as well in the orbital frontal area. We move from gustatory to vision. We'll talk about a few terms you're probably very familiar with already, but just for sake of completion, we'll go through them again. So vision basically talks about the wavelength of visible light that we can perceive by sight, which is going to be electromagnetic spectrum between ultraviolet rays and infrared rays. So all the colors of the rainbow, word GBIV that you remember from physics class back in high school, these wavelengths will go from 400 to 700 nanometers. And of course, the measure of the nanometers is in relation to the actual wavelength of the type of light in the electromagnetic spectrum, which is basically defined as the distance between 2 consecutive peaks of electromagnetic wave. And of course, closer to the violet end of the rainbow part of the spectrum is going to have a shorter wavelength than, say, the red area of the electromagnetic spectrum, which have a longer wavelength. The accessory structures around the eye include the eyelids, the eyelashes, eyebrows, your lacrimal ducts, the tear-producing apparatus, and also the extrinsic eye muscles. The palpebral muscles are going to control eyelid motion, and the extrinsic eye muscles will be responsible for the actual movement of the eyeball itself. You have the conjunctiva, which is the thin protective mucous membrane. It lines the eyelids and covers the sclera. Those of you who have gotten red eye before, that's also known as conjunctivitis. So it's inflammation of this mucous membrane. We also have a tarsal plate, which is basically a row of mucus glands, always known as meibomian glands, that basically when you close the eyelids, it prevents the 2 tarsal plates, the 2 eyelids, from connecting to each other. Now, some things you'll see if there's really bad conjunctivitis or any kind of inflammation of the meibomian glands, overnight you may actually start to secrete mucus. It'll harden overnight. I know this happened to my kids as well as some other patients' kids, where it's almost the mucus can harden so hard that you can't open the eye, which can be somewhat scary for a young kid not be able to open their eyes upon waking up. Fortunately, though, using just a hot washcloth or anything with a little bit of moisture to it, you can basically moisten up the mucus to allow the eye to basically to open up again. But generally, the meibomian glands, if they're processing the right amount of mucus, it basically allows the tarsal plate to basically slide on one another and therefore be able to open up the eyelid. And here's a sagittal shot of the left eye. So basically what you're looking at here, you start in the posterior, you see the optic nerve, which we'll get to in a little bit. You see the superior rectus and inferior rectus muscles, basically going from anterior to posterior. See the levator palpebrae superioris muscle, which will raise the eyelid. You can see more towards the anterior portion, you can see the eyelashes. You can see the edge of each of the eyelids, both the lower and upper eyelids or palpebra. At the very tips of those, you'll find the meibomian glands or tarsal glands right there in green. These are responsible for secreting the mucus to allow the eyelids to separate appropriately. Look at some of the other things a little more closely on a different slide. You can see the pupil, the lens, and the iris. You can see the different types of the canal of Schlemm, which we'll get to in a little bit. You can look at the different functions of the eye here. Notice how basically the shape of your eyeball is going to depend on the eye orbit, which will basically dictate what kind of sight you may have, whether it be normal sight, nearsightedness, or farsightedness. So the lacrimal apparatus we're going to talk about with the different types of functions, as well as how tears actually form and where they are drawn from. So very simply, you're going to have the lacrimal glands, which is going to be the most superior and lateral portion of the lacrimal apparatus. These will secrete the actual tears. They will in turn dump into the lacrimal ducts and go into the lacrimal puncta. And then once you get into the lacrimal puncta on the medial side, it'll drop down into lateral canaliculi, and it'll dump itself into the lacrimal sac. This is going to be just superior and lateral to the nose, and this will also feed into the nasal lacrimal ducts that flows the tears into the nasal cavity. Now this makes for a great test question. This gives you the actual flow of how tears will go from one part of the apparatus to the next. It starts in the lacrimal gland where the tears will be secreted. It will dump into the lacrimal ducts, and from the lacrimal ducts they'll spread over the entire surface of the eye. So not only will they drop inferiorly, but also drop from lateral to medial at the same time. These will further flow into the superior and inferior lacrimal canaliculi, which will further drain into the lacrimal sac, and then back into the nasolacrimal duct, which will further drain tears into the nasal cavity. So basically, as the tears will flow, they tear into where the lacrimal puncta is, and also to the lacrimal caruncle, as it goes into the canaliculi as well as the lacrimal sac. So two things of note here. One in particular, this is why anytime you cry, whether it be from emotional crying, whether it be biting something spicy, or whether it be actual injury, this is why your nose gets sniffly after you have a lot of tears, because it dumps right into the nasal cavity. The second thing is more so for those of you who are into acting in particular. If you notice the flow of the tears, they go from the lacrimal duct, the gland to the duct, and then travel from the lateral to the medial. You see people when they tear up naturally, the tears will flow around lacrimal puncta as well as the lacrimal caruncle and flow downward from there. So it goes down the medial end of the eye. If it goes down the lateral side, it's usually what we call fake tears. You see it a lot of times in different movies and different TV shows, usually from eye drops, that kind of thing. But normally tears will flow from the medial side of the eye and down towards the nose. And of course, if you remember from the muscle system chapter we talked about back in chapter 11, there's going to be 6 extrinsic eye muscles. We have the 4 rectus muscles, superior, inferior, lateral, and medial, and 2 obliques, the superior and the inferior. Now, also covering the eyeball, you're going to have 2 different types of tunics or coats. You have the fibrous tunic, which is going to include the cornea and the sclera. So the cornea is going to be the outermost portion that faces the outside environment. The sclera is going to be the outer white portion of the eyeball. And then contained therein is going to be the vascular tunic, which is going to include the choroid, the ciliary body, as well as the iris. So back to the slide again, you can see 4 of the extrinsic muscles. 2 are missing here just because of the sagittal section taken. So the medial and lateral rectus cannot be seen. You can see the superior and inferior rectus as well as the superior oblique as well as the inferior oblique muscles as well. You'll also see the levator palpebral superioris muscle that helps open the eyelid. Now, as far as vision goes, this is probably the most important slide of the vision portion of this chapter, and I know there's a lot to take in. So basically try and break this down a little bit here. So first, if you look at the different types of tunics, so the fibrous tunic, if you look at the cornea, if you follow the shaft of the light, the cornea is going to be continuous with the sclera, the white portion of the eyeball. The vascular tunic is going to be made of the choroid, the retina, as well as the iris in particular. So that takes care of the outer coverings of the eyeball. If you go just interior to the cornea, you're going to find 2 chambers. You're going to find the anterior and posterior chamber, which we'll get into in just a little bit. Very simply, you follow the shaft of light. It'll pass through the pupil, which is the opening within the iris. This can dilate as well as constrict. Next, you have the lens, which is made up of crystalline liquid or proteins. This has the capability of flattening or actually thickening, going into concavity or convexity, to basically accommodate vision both near and far. This is helped by the zonular fibers that are going to be attached just on the periphery. This will also be attached to the ciliary body, which is going to include the ciliary muscles and the ciliary processes. So these help accommodate the lens in particular. The iris, as we talked about before, this iris is actually made up of different types of muscles, which we'll get into a little bit. This will open or close the actual pupil area to allow a certain amount of light to come in. Now, if we follow the light as it passes through the anterior and posterior chambers, before we get to the back of the retina, it's going to pass through or pass by the oris serrata, which is going to be, if you remember the serratus anterior muscle, it's going to have a very sawtooth-like appearance. This anchors the iris to the retina and to the sclera. So if you follow the light further back, let's get into where you see both the macula lutea and the fovea centralis. The macula lutea is nicknamed the yellow spot. And deep within the yellow spot, you're going to find the fovea centralis. And this is important because this is going to be the highest area of visual acuity. This is where you're going to have the highest amount of cones, which are going to be for detecting colored light. So these are going to be found in three types: red, green, blue, which we'll get to later on. And interspersed within the fovea centralis are going to have a lot of rods, which are for bright light. And then when you go into the rest of the retina, they have more rods than cones, more for detecting bright light, not so much for actual color. Now, just medial to that, you're going to find the optic nerve, as well as the optic disc. This is where you can find the optic nerve, the central retinal artery, and vein all mixed in together there. This was called which is known as your blind spot. There are no photoreceptors or any receptors for light in this particular spot. We get into the lab 13. We'll be testing your blind spot in very simple exercise. We'll talk about that later on. Other than that, I want to point out too, you'll see the lacrimal sac just medial there. You see the canal of Schlemm, which actually help lubricate the mucosal line, help lubricate the area, as well as keep a healthy environment for the anterior and posterior chambers. Here we have two different types of muscles found within the iris. You can find both the sphincter and the dilator pupillae. So these are based on the autonomic reflexes. These have direct input from the autonomic nervous system, both the parasympathetic and the sympathetic. So the parasympathetic will deal with the sphincter pupillae, which will decrease the pupil area to allow a little bit of light to come in. So we find this especially in bright light. We also find this when we're very relaxed. On the flip side, if there's very dim light, or very dark in a dark environment, the pupil will dilate with the dilator pupillae. These is tied in more so the sympathetic nervous system. So we find this if you're in fight or flight, you want to have the pupil dilate to see as much as possible in a split second. But there are also some artificial ways to both constrict and dilate the pupil. Those who've been to the optometrist before, you've most likely had your pupils dilated by different eye drops. This basically will stun the dilator pupillae, or basically will anesthetize it. So basically, it will not function anymore. I'm sorry, it'll actually inhibit the sphincter pupillae and actually will activate the dilator pupillae. Pardon me. Other ways that can also dilate too is if you're on certain types of drugs. Usually hallucinatory drugs will actually cause the dilator pupillae to actually constrict again and open up the pupillar area. So again, using a fundoscopic exam using an ophthalmoscope, you can actually look inside the eye through the pupil, through the iris, to get an idea of what the actual retina looks like. The retina itself takes up about 3/4 of the inner layer of the eyeball. You can see the optic nerve. Generally, you see the optic nerve will have different arteries that will sprout from it, and then usually just medial to that, you're going to find the actual fovea centralis and the macula lutea, which is going to be the yellow spot. But the optic disc, again, has no cones, no rods, so there's no receptors found at the blind spot. But the area of highest visual acuity, known as the fovea centralis, is basically located within the yellow spot called the macula lutea. So the first of our critical thinking questions is going to involve the fovea centralis. So again, it's going to be the area of highest visual acuity. It's where you're going to have, in particular, I'll give you the answer right off the bat, going to have the highest concentration of cones, and cones are found in 3 different colors: red, green, and blue. It will be interspersed with some rods too, but the cones in particular will dominate the area of the fovea centralis. The rods are actually located more so on the periphery of the retina. You'll still have some cones that will spread throughout the retina as well, but the highest concentration will be found in the fovea centralis, with the majority of your cones there. So this is why when you look at a particular area, out of the corner of your eye. When you actually focus on it, you can actually differentiate different colors a lot more easily than if you're looking from your periphery. Now on the flip side, now at night when you're viewing a star, a lot of times if you look at a star off an angle, just off your center of the focus centralis, it'll look brighter because you have more rods in particular that are viewing the star for brightness sake than you are for color. But when you go to look at a star directly, it seems dimmer. So the whole reason being is the focus centralis, again, will have a higher concentration of cones. So it's very good for visual acuity and for recognition and to differentiate different colors, as well as in conjunction with the rods for visual resolution. In other words, be able to differentiate one particular border or one particular part of something versus another, so it's not very fuzzy. On the flip side, though, at night, though, you generally have more rods that are firing off than cones. This is why you can't see color too well at night, but you can see different amplitudes of brightness in particular at night. But again, even looking off center will actually give you better brightness than if you look right at a particular star in particular, because you have not as many rods that are activating right at the focus centralis. So using an ophthalmoscope, you can do a fundoscopic exam where you basically look through a person's pupil and see this image through here. So some areas of notice will be the optic disc found on the nasal side here. So you notice the deepest portion of it is very white, a little yellowish towards the edge. You can see the retinal blood vessels that will branch off there out. So these are arteries and veins that branch off the optic disc. You go more towards the lateral side, almost dead center, you're going to find the macula lutea, which is going to be the yellow spot, and deep within that is going to be what's known as the fovea centralis, where it's going to be the highest area of cones in particular to give us the highest sense of visual acuity. Naturally, there are two types of photoreceptors we find in the eye, or the rods and cones. Rods for dim light, cones we use for color vision, and we have three different types of cones: red, green, and blue. From these sensors, information is going to flow out through the outer synaptic layer to the bipolar cells, to the inner synaptic layer to the ganglion cells, and all these will actually flow through the optic nerve. So if you look through here, very simply, if you go from the retina and then work from the superficial to the deep, you see the ganglion layer. We find the ganglion cells, followed by the inner synaptic layer, and the bipolar layer, which you'll find the horizontal cells, the bipolar cells, and endocrine cells. These cells are more inhibitory. These are very important for trying to maintain resolution, or the ability to differentiate one object from another. You can notice also have the outer synaptic layer followed by the photoreceptor cell layer, which is where you'll find your rods and your cones. The inner segments you can see where the majority of the cell body and cell nucleus. The outer segments will find the different rods and cones. So if you notice here, the impulses of the nerves will travel from the outer segments to the inner segments, through the photoreceptors to the outer synaptic layer, out to the ganglion center, and then out to the optic nerve axons. The photons of light travel just the opposite way, so they'll actually run through the retina, past the ganglion cells, past the bipolar layer, to the actual rods and cones. We see the little foldings there, which we'll talk about in a little bit. So it's going to pass through all these structures to strike the rods and cones. So if you think of a camera in particular, it's almost as if you're putting all the wiring of the camera ahead of the film. So from an evolutionary standpoint, this is how the eye has developed, which is somewhat backwards than we would normally think, but this is what we have based on the evolutionary process. Here's a nice little sagittal shot of the optic nerve. So this is the reason why we call this a blind spot. The optic nerve will project through the retina. If you notice on either side of the optic nerve, you can see rods and cones all throughout the retinal layer. There's nothing there at the optic disc, so there's nothing to detect any kind of light. So deeply embedded within the optic nerve, you find the central retinal artery and central retinal vein that give you the retinal blood vessels. We also have different chambers that are going to be fluid-filled different portions of the eye. You've got the anterior and posterior chamber that's going to be segmented by the iris. The anterior chamber is going to be filled with what's called aqueous humor. This is a very clear watery liquid that's replenished every hour and a half. You also have the posterior chamber, which lies just behind the iris and from the lens, also filled with aqueous humor as well. Now, just behind the posterior chamber, you find the posterior cavity. So you fill with the vitreous chamber or vitreous fluid. This is also transparent, a little more gelatinous. It's not readily replenished, and actually we use the vitreous humor to actually keep the eyeball in a spherical shape. Those people that have injuries to the eye where the vitreous humor has leaked out, very simply can cause the eyeball to collapse. On the flip side, if you have too much vitreous humor, this actually causes glaucoma, too much pressure on the retina. And if you remember back when we talked about having too much CSF in the meninges, if there's too much pressure there, it can actually cause some necrosis because it shuts down the blood flow, so it can cause those cells to die. So very simply the same kind of thing you can see with too much vitreous humor or too much pressure found in glaucoma can actually cause irreparable damage to the retina. So here's a nice little shot, a sagittal plane shot of different chambers found in the eye. So again, the anterior cavity is made up of two chambers. You have the anterior chamber found just deep to the cornea. It's going to be separated by the iris here. The posterior chamber is going to be found between the iris and the lens. Found just posterior to the lens, you find the vitreous chamber filled with vitreous fluids. Now, if you notice here, there's a certain type of flow that happens in the aqueous fluid in the anterior and posterior chamber. Basically, the arteries and veins from the ciliary area here will flow through the posterior chamber and be circulated through to the anterior chamber, and it will be drained through the sclerovenous sinus. This table here gives a nice quick overview of the major structures of the eye. This is a great little snapshot of what you need to study for the final exam. So let's talk about how the light passes through and actually strikes the retina. So as we know, light has to pass through the clear surface of the cornea. It'll pass through the clear fluid of the anterior chamber, pass through the opening known as the pupil. It'll pass through the posterior chamber, through the lens, through the vitreous humor, and then finally it'll project onto the retina. So as we know from physics back in high school, light will refract or bend when it passes from one substance to another, when it has different densities between the two substances. It's this bending that allows us to actually detect certain convergence of light on retina and basically to see a very distinct image. Now the images in particular, when they are flashed onto the retina, they're going to be inverted and right to left reversed. So complete opposite what we normally see or normally would expect. Now the brain will autocorrect for this image, also using different types of input. So very simply using touch will give us an idea of how to revert the image back the correct way. We've learned this through evolutionary practices. Also the lens will learn how to accommodate, to properly focus on an object. Basically it can accommodate up to about 10 centimeters. After that, very simply, there's no amount of accommodation that will allow the image to be sharp and focused. That's why when you bring something too close to the eye, it gets very fuzzy, it gets very blurry. Ideally, the image is going to project right on the central fovea, or the fovea centralis. This is where your vision is going to be the sharpest, so we have the best amount of resolution. So here we show the vision of accommodation. This is where the lens will actually change shape to accommodate the light rays from a distant object. So on the left side, we see the viewing of a distant object. We see the light rays will bend as they pass through the cornea and the aqueous humor. It'll bend through the lens again to allow a nice sharpening conversion point right at the retina. In this case, when we're looking at a distant object, the ciliary muscle is relaxed, so the zonular fibrils will remain taut and give you a nice flat lens. Conversely, we have a close object, the lens becomes more spherical, where the ciliary muscle will contract and therefore loosen the zonular fibers. This will sharpen the angle a little bit and allow the conversion point to rest on the retina appropriately. Now normally, when the conversion point will rest on the fovea centralis appropriately, we have what's known as an emmetropic eye, where very simply the light will correctly focus on as a clear image onto the retina. Now when we have nearsightedness, this was known as myopia, where very simply the eyeball is too deep or is longer than it should be, and basically the convergent point actually focuses just in front of the retina. This is why a lot of times you'll actually see a image that's very blurry. We use a concave lens used to correct the vision. So basically we're going to expand the angle before it hits the lens to basically lengthen the point of convergence of the light. This can be fixed by concave lenses as far as eyeglasses go or as far as contacts go. So here we see on the left side, we see an uncorrected myopic eye, where very simply the plane of focus is going to focus just ahead of the retina and gives a very blurry image. So we use a concave lens that's going to expand the refraction, or it's going to change the angle as it passes through the cornea, as it goes through the lens, and therefore let it fall right on the retina where it should be. Now we have farsightedness. This was known as hyperopia or hypermetropia, where very simply the eyeball is shorter than it should be, and the image will converge just past the retina Usually a lot of times will fall on the sclera or even past that. So usually for distant objects we can see those relatively clearly, but again we have difficulty seeing close objects. So in opposition to using a concave lens, we use a convex lens to correct farsightedness. So here we see a farsighted eye that's uncorrected. We see the angle of the conversion point extends far beyond the retina. We use a concave lens. We'll sharpen the focus of the light rays to allow the conversion point to rest right on the retina. Now, with both of these different types of eyeballs, it's based on the shape of the eye orbit of your skull. So a lot of times it's going to be influenced by genetics. So more often times than not, if one or both of your parents are either nearsighted or farsighted, odds are you'll end up nearsighted and farsighted just like they will, unless you happen to get a different shaped skull. Very simply, the eye grows into the eye orbit and takes the shape of the eye orbit. So if it's too elongated, like we saw in nearsightedness, you're stuck with nearsightedness. If the eyeball is too shallow, then you're stuck with farsightedness. Now, a different type of visual distortion that we see is what's known as astigmatism. Normally, your cornea and your lens will have a uniform curve all throughout, but occasionally you may be born with what's called astigmatism, where there's an irregular curve. So most oftentimes, optometrists will signify a certain astigmatism along a certain plane across the eye. More oftentimes than not, they use the hands of a clock. So very simply, you might have an astigmatism that runs from 12 to 6 o'clock or from 1 to 7 o'clock, or in some very rare cases it may be distorted, where I say it's from 1 to 5 o'clock. So these a lot of times can be corrected by eyeglasses by changing the shape of the lens. For contact lenses, a lot of times these are weighted, so very simply when you put the contact lens in, it will always orient itself in the right position to basically correct the astigmatism. Some people nowadays will offer to take LASIK, which is laser-assisted in situ keratinolysis. Years ago, back about 20 years ago, this was done by a scalpel. Nowadays they use this by laser. They've gotten very good at doing this. In years past, when it was first developed, they noticed a high correlation of cataracts with people that have had this done. Nowadays they can correct this very quickly and very easily. You are awake for the test. What they do very simply is they will numb the eye. They'll use lasers to basically laser off the cornea. They'll reshape it and then basically fuse it back onto the eye. Generally, you wear an eye patch for about 24 to 40 hours. You take lens wetting drops, and usually you achieve somewhat 20/20 vision very soon thereafter. Most people get one eye done, and then within a couple days or a few weeks, they'll get the other one done once the other one is fully healed. Now, two other aspects of our vision that's very important. Two terms we use is called convergence and binocular vision. So very simply, you compare us to a animal such as a bird or horse. Very simply, they have one eye on either side of the head, so each one has its own field of vision. We have something very different where both eyes are set in the forward set position, so we have what's called binocular vision. So this allows us to converge, or very simply have both eyes focus on the same object at the same time. Also, we have fields of overlapping vision. This gives us the ability to have depth perception. So it's that slight differential from one eye to the other as far as shadows fall that gives us an idea of how things are spaced. So how far things may be gives an idea what depth perception is. And those of you who have ever driven before, playing that kind of sport, you know how important depth perception can be. For a little trick, you can actually practice trying to drive in a safe area course with one eye. You notice how difficult it is to perceive depth perception with a single eye. So here you see a close-up of both the rods and cones. These are two photoreceptors found in the eye. On the left-hand side, you can find the discs here of the rods found in the outer segment. You can find the folds in the outer segment of the cones in particular. So very simply, they're simply named by how they're shaped. So to achieve a receptor potential, all your rods and cones will contain photopigments. Now, there'll be different types of photopigments found, but for now, we're only going to learn about the rods. We'll have a photopigment known as rhodopsin. The cones have their own different types of photopigments. They're going to be signified by the red, green, and blue types of cones, but all of them will follow response to light in a cyclical process. So left here you can see the photopigment rhodopsin is going to be made up of two types of proteins. One is retinol, one is opsin. You'll find these also in the folds found in the cones as well, but all of them will follow the same process. So very simply, we have cis-retinol in the colored photopigment. When it comes in contact with a photon of light, it'll isomerize or straighten out. The retinol will start to detach, and very simply, the retinol in particular gives the particular photopigment its color. When the retinol is detached, this is known as bleaching. So very simply, it loses its color. So very simply, the opsin will stand free. Retinol isomerase will convert the trans back to cis-retinol. The free-floating cis-retinol will bind again with opsin, and we're back to this initial stage, ready to accept another photon of light. So we have different types of adaptation processes, both light and dark. So for light adaptation, it's a very quick process, very simply because the amount of time it takes for the retinol isomerase to actually regenerate the retinol back into its cis form is happening very rapidly in the cones in particular, very simply because you have a lot of cones that are for different types of wavelengths of light. So again, they're dedicated, they're either red, green, or blue. So you have a lot more available that can adapt to light very quickly. So in other words, you go from a dark environment to a light environment, you can adapt to light vision very, very easily. Now conversely, for dark adaptation, because rods in comparison to cones do not regenerate very quickly, very simply because there's not as many available, it takes a little more time for you to adapt to dark light. So very simply, you're in a light room, you shut off all the lights. It's going to take a few minutes. For some people, it can take anywhere from 5 to 8 to 10 minutes or so to fully get your dark vision back. Very simply because the cones are basically inactive. There's not much they can really do to aid in dark adaptation. So the rods are pretty much your primary photoreceptor in dark light. So very simply what ends up happening is the light in the photoreceptors, we'll talk about the neurotransmitter glutamate. Very simply, when we talk about the bipolar layer of the retina, very simply, the inhibitory transmitter glutamate will turn these off to allow more rods to fire off to at least give you as best acuity as possible or best resolution possible in dark adaptation. And one more thing I want to add too, if you also know, especially when you're in a dark room, you can't distinguish color very well because again, there's not enough cones being fired off to be able to differentiate different types of color. So this is going to be a very brief overview of how in light in particular, what happens with the isomerization of retinol and cyclic GMP. So very simply what ends up happening is it causes a hyperpolarizing receptor potential. So very simply in light, the glutamate release is turned off. This will excite the bipolar cells. So very simply what happens here is when you have a lot of bright light, there's a lot of light coming through the pupil, it's going to fire off a lot of cones. So very simply, to basically sharpen your focus and sharpen resolution, you want more glutamate turned off to allow the bipolar cells to basically sharpen the focus for all the nerve impulses traveling to the optic nerve to the brain to give you a sharper image. Now on the flip side, for photoreceptors in darkness, very simply for the rods, you want to inhibit the glutamate because you want to have as many of the rods firing off as possible to generate as much of an image as you can. So very simply, in this case, these will depolarize to allow for the full action potential to occur in the photoreceptors in particular. So again, in this case, glutamate is being is going to be inhibited to allow, is going to be released more so, to inhibit the bipolar cells to give you as much of an image as possible. It may not be sharp. That's why our acuity is not that great, especially in darkness. So you want to try and get as much input as possible. So the neural pathway in particular, so if you take in the image of light and how it's transmitted to the optic nerve, it's going to follow this particular pathway. The only area you may be familiar with already is the optic chiasm. We looked at the cranial nerves, but basically it's going to partition from the optic chiasm to the optic tract. It's going to synapse into the thalamus at the lateral genicular nuclei, and then it's going to finally go to the optic radiations. It's going to go to the primary visual areas to the occipital lobes. Now it's important to remember the occipital lobes actually get the image, but the recognition of the image goes to the frontal lobe of the cerebral cortex. So here we see the inferior view of the brain. So again, the visual pathway will start off at the optic nerve, follows to the optic chiasm. Portions will go to the same side of the brain, some will go to the opposite side of the brain. It'll flow through the optic tract, synapse at the thalamus at the lateral genicular nuclei. It'll follow out to optic radiation, which should look very familiar to, should look very similar to the white commissure tracts we found in the corpus callosum, and finally ending up in the occipital lobe at the primary visual areas of the cerebral cortex. So again, as I mentioned before, it's the anterior or front-loading vision of the eyes that gives us our visual field overlap, gives our binocular vision. when you do the test for the labs, checking for the blind spot, very simply, if you had just simply one eye, if there's one particular portion of your eye that does not pick up any light, that's going to be the blind spot where the optic nerve is going to emerge. We do not normally see our blind spot because the other eye will compensate for that particular blind spot on the opposing eye. This is a very important thing for binocular vision. So not only does it compensate for the optic nerve on both sides, the quote-unquote blind spot, but also helps us differentiate different types of 3D images based on differences of shadows from one eye to the next, that change in angle from one eye to the other, that gives us our depth perception. So again, we're going to follow through. So you see the overlapping vision here. The visual field left eye overlaps the right eye. If you notice, it's not complete overlap, very soon because the bridge in the nose is going to block one eye from the other, getting the full field of view. And we'll go a little more closely into which part of the visual view goes to each part of the brain where it ends up. Some do cross, some do not. So basically we have two visual fields of each eye. We have nasal and the temporal. So very simply, it would be medial and lateral. So the easiest way to remember it is remember which portion of the eye goes to which portion of the brain. It follows like this. The right half of each visual field, so each eye, the right half of the left eye, right half of the right eye and left eye go to the left side of the brain. Conversely, the left half of each eye, of the left and right eye, goes to the right side of the brain. And this is what it looks like. So again, if you just simply remember that both left halves go to the right side of the brain, and both right halves of each eye go to the left side of the brain, this is actually very simple to pick up. So very simply, if you look here, the temporal half of the left eye and the nasal half of the right eye end up on the right side of the occipital lobe. Conversely, the nasal half and temporal half, nasal half of the left eye, temporal half of the right eye, notice from the right side, they will end up on the left occipital lobe. The final section for special senses will deal with hearing and equilibrium. Now, for hearing in particular, or when we use transduction of the sound vibrations, for the ear sensory receptors, these are going to be vastly faster than the photoreceptors found in the eye, about 1,000 times faster in particular. This is very important in particular when you do the special senses lab. If you notice when you do the ruler test, if you do the ruler test just by sight, by having someone grab it, first when you give them a command word, a very peculiar thing happens here. Some people think by having two different types of stimulation will slow things down because it's a distraction to the particular person, but actually what winds up happening is because we respond more quickly to the transduction of sound vibrations, you can actually catch the ruler a lot more quickly than you would with the command word versus just going by sight. So it's a very valuable thing for you to do when you're looking at the special senses lab. Hopefully you've looked at my narrated PowerPoints first, because that very simply gives you the answer why having command word actually will decrease the response time and give you a better response time when you guys are practicing catching the rulers. Now, besides detecting the transduction of sound vibrations, it'll also be very important for the receptors for equilibrium found in the inner ear. So the ear is going to be divided into three regions. You have the external ear, the middle ear, also the internal ear. So here's a nice little representation of all three partitions. So let's start from the periphery and work our way more deeply. So we have the auricle of the ear, very simply shaped somewhat like a cone. You have the helix, which is the upper portion, and the lobule, which is a little dangly portion that most people get pierced. You have the external auditory canal, also known as the EAN. This is where your earwax or ceremonious glands will actually secrete the wax-like substance, help basically cleanse the area and trap any debris that might travel in through the ear. Very simply, it's going to be encased within the temporal bone. And the last partition you'll see there is the tympanic membrane, otherwise known as the eardrum. That's going to be the entirety of the external ear. So you look at the middle ear, you're basically looking at a couple different structures here. You're looking at three smallest bones you're going to find in the body: the malleus, the incus, and the stapes, otherwise known as the hammer, the anvil, and the stirrup. These bones, will vibrate very frequently. That will amplify sound as it goes in towards the inner ear, which you're going to see very peculiar structures there. You're going to see the oval window, which comes right up against the stapes in particular, as well as the round window. You're going to find the auditory tube. This is what's known as the eustachian tube in old school vernacular. Normally, the eustachian tube for most adults will actually travel from the superior to the inferior, and also go from posterior to anterior, and also travel lateral to medial. In kids, these are not angled quite as sharply, so a lot of times they'll get some backup of mucus in through here. So a lot of times they may need to get their ears drained. Some people have actually gotten tubes put in the ears to allow proper drainage of that particular area. If you go a little more internal, you find the cochlea, which is very important for detection of vibrations of sound through sound waves. You have these semicircular canals, which are going to be at 90 degrees to each other. This helps us work for angular acceleration. Also, you're going to find the utricle and the saccule found in between there, and all these will synapse with different portions of the vestibulocochlear nerve, cranial nerve 8. You have both the vestibular branch for equilibrium, the cochlear branch for sound. So again, the external or outer ear is going to contain the auricle or the pinna, the external auditory canal, as well as the tympanic membrane or the eardrum. So the auricle will basically help funnel sound as it travels through the external auditory canal. Basically, it will transmit to the eardrum, and of course, the ceruminous glands will secrete the cerumen or the earwax to protect the canal from debris. The middle ear had 3 small bones in the body called the auditory oscles. Again, the malleus, the incus, and also the stapes. So these sound vibrations are going to transmit through the eardrum to 3 bones to the oval window, in which the stapes is going to lay into. The auditory tube will extend from the middle ear to the nasopharynx to regulate air pressure into the ear. A lot of times we'll use this to help equalize pressure in the ear, especially when we're taking flights in high altitudes, and also when we descend. Now, aside from the 3 smallest bones we find in the body, we also find 2 very small muscles in through here as well. Now, these are both going to be involuntary muscles. You have the tensor tympani muscle, which is going to be innervated by the trigeminal nerve, and then the smallest muscle you're going to find in the body, known as the stapedius muscle, is going to be innervated by the facial nerve. Both of the muscles will help dampen any kind of tension on the tympanic membrane, especially in the presence of loud noises, so therefore it doesn't cause any kind of damage. This will absorb some of the vibrations to kind of soften the blow, so to speak, from any type of either loud noises or very high frequency noises. The inner ear, or the labyrinth, contains the cochlea, which is going to transmit vibrations and neural impulses in the brain. There's called semicircular canals, and also works with the cerebellum for balance and equilibrium. So it's going to have a special kind of fluid found within the cochlea and semicircular canals known as perilymph, which is going to be somewhat similar to cerebrospinal fluid, except it's going to have a lot more potassium found within the fluid itself. So here you see the components of the inner ear. Let's look at the anterior portion first. You're going to find the round window where the stapes will be just superior to that. Basically, it's going to allow proper flow in through the cochlea and the cochlear duct, so it's going to wrap around almost like a snail's shell, so to speak. And it's going to have 2 different partitions to it. So it's going to detect different sound waves as it transmits through the perilymph, and it's going to detect those 2 changes as one flow of the perilymph goes in one direction, one goes in the opposite direction to help maintain or perceive different types of sound. And we'll get into more so of the posterior area in just a few minutes. Very simply, the areas we'll look at are the utricle as well as the saccule. These are going to be very important for what we call linear motions. And then you also have 3 different semicircular canals, all at 90 degrees. You have the anterior, posterior, and lateral. These are for angular acceleration. Very simply, these are very important for angled motions. So if you can think of it this way, where linear motions would be simply going up and down an elevator or walking across a flat surface, whereas angular acceleration would be if you're simply turning to one side, tilting the head to one side during flexion or extension or hyperextension or lateral flexion or rotation. So the vibrations as it passes through the tympanic membrane, it's going to flow through the 3 different auditory oscles. So it gets to the stapes up into the oval window. So what winds up happening is each of these structures gets smaller and smaller, and the density will actually change a bit to allow the vibrations to become 20 times more vigorous than those at the tympanic membrane as it passes through the oval window. So basically what ends up happening is these sound waves will pass from the cochlea into the fluid as pressure waves that gets transmitted through the perilymph as what's called the scala vestibuli. So from here, the pressure waves from the scala tympani and then to the round window, which is going to bulge into the middle ear. So here we see the sound transmissions from the stapes at the oval window as it travels to the scale of vestibuli. It's going to follow around through the cochlea as it's going to transmit at the helicocentrema, which is going to turn around very simply, going the opposite direction, and then basically spin out to the scala tympani. And very simply, it'll end up at the round window, which is going to be covered by a secondary tympanic membrane, which basically will end where the sound waves will terminate. Here's a cross section of one turn of the cochlea. So as you see here, the scala vestibuli is going to be just superior to the scala tympani. It's going to be the change in the different types of pressure waves from each of the different scala there. It's going to be detected by what's known as the spiral organ or the organ of Corti. Basically, this kind of vibration will cause a tugging of the tectorial membrane, which will activate some of the neurons found deep within the spiral organ, which will synapse with the cochlear branch of the vestibular cochlear nerve. So the pressure waves there, again, we're going to travel from the scala vestibuli to the vestibular membrane into the endolymph of the cochlear duct. So what happens, the basilar membrane is going to vibrate. So specialized hair cells, the spiral organ, against the tectorial membrane will vibrate and basically depolarize. So here we see a close-up of the tectorial membrane. You can see the specialized outer cells from the basilar membrane and also the inner hair cells through here. So very simply, it'll cause a mechanical change to the stereocilia, and we'll see in the next slide or two how different sodium ions will rush in and actually will mechanically open up those gated channels to allow depolarization. As you can see, it'll synapse, especially the inner hair cell, will synapse with the sensory portion and the motor portion of the cochlear branch of the vestibulocochlear nerve. So it's this input from the motor fibers that helps adjust the tension at the basilar membrane. So here's a quick little overview of the sound waves that travel from the external auditory canal. It will strike the tympanic membrane, cause a vibration with the auditory ossicles as the stapes attaches up to the oval window. It'll cause pressure waves to the perilymph in the scala vestibuli. As it travels through and ends up going in the opposite direction at the helicocentrema. And as it passes through all these areas, it's going to be detected both in the spiral organ of Corti, also through the cochlear duct. And finally, it's going to travel through the scala tympani. And again, as it passes through this whole area, the organ of Corti is going to detect the two differentiation of the pressure waves from both the scala vestibuli as well as the scala tympani, and finally end up at the secondary membrane at the round window. And here we see the synaptic points for the two portions of the vestibular cochlear nerve, with the cochlear branch and the vestibular. So obviously the cochlear branch is going to synapse at the cochlear area as it branches off to each of the organs of Corti. You're going to see the other branch, the vestibular branch, is going to branch off to the saccular as well as the utricular nerves, as well as the ambulatory nerves, which we'll get to in just a little bit. So cochlear nerve branch will be the cranial nerve 8 from the vestibulocochlear nerve. The axons will synapse with neurons in the cochlear nuclei in the medulla oblongata. The impulses will travel to the medial geniculate nucleus of the thalamus and goes to the primary auditory area at the cerebral cortex, again in the temporal lobe. So here we find the hair cells that can be found in the organ of Corti as it attaches to the tectorial membrane and actually will synapse with the first order neurons found in the cochlear branch of the vestibulocochlear nerve. So very simply, the image on the left here shows a resting hair cell where all the potassium ions are weakly open. Very simply, the sericilium to improve surface area for the potassium to rush in. When we get any kind of vibration or any kind of tugging on the sericilium, these will mechanically open up the potassium channels and allow potassium to flow in, therefore depolarizing the hair cell. This will release the neurotransmitter, synapse the first order auditory neuron, and then achieve the depolarization for the neuronal impulse. As you see the pathway here, you see it's going to synapse both on the same side as well as the opposite side of the cerebral cortex. It's going to synapse at the medulla oblongata, at the cochlear nuclei, as well as superior alveolar nucleus in the pons as well. It'll synapse through the thalamus at the medial geniculate and go out to the primary auditory cortex in the temporal lobe. Now let's talk about the vestibular function of the cranial nerve 8, or for vestibular function as far as equilibrium goes. Equilibrium in particular means balance. It exists in two forms. We have static and dynamic. Static is very simply trying to maintain the body's position in any position relative to the force of gravity. And this could be any position. It could be standing, it could be sitting, it could be laying prone, supine, what have you. Dynamic equilibrium is the maintenance of the body's position in response to sudden motions or fluid motions. There are different apparatus found here, found deep in the inner ear, known as the vestibular apparatus. So these particular organs are going to help maintain your equilibrium both from a static and also dynamic position. You have the saccule and the utricle, which are known as otolithic organs. Very simply, your saccule is what detects vertical motion. So think of being in an elevator. It detects whether you're going up or whether you're going down. It's going to be that relative change of position that it detects, which we'll get into in a few minutes. Your utricle is going to be more so for horizontal positioning. So in this case, if you're in any kind of motor vehicle, whether it be your own car, or whether you're on a bus, train, you're in a plane, it's going to be that feeling you get when the sudden acceleration or deceleration, the quick acceleration when you feel yourself getting pushed into the seat, or the quick jerky motion when you go forward, that is detected by the utricle, is going to be horizontal acceleration or deceleration. The semicircular canals, these are going to be formed at 90 degrees to each other. You have an anterior, posterior, and lateral. These are going to detect annular acceleration. So if you think of all the different ranges of motion we checked on during the joints lab, it's basically angular acceleration is going to be around a certain pivot point. So if you think of all the head motions, whether it be flexion, hyperextension, rotation, lateral flexion, these are all angular accelerations. Now, the way that we detect these is through the use of otoliths, which are going to be calcium carbonate crystals. These will sit deeply in what's called the macula. So very simply, or actually very similar to the types of hair receptors we had back in the ear for hearing, they use something very similar to this for equilibrium and for maintaining positioning. It's very important, in particular, for static equilibrium into maculae for the saccule and the utricle. Now, very similar to the tectoral membrane, we have an otolithic membrane that sits on top of the macula. So this is where movement of the head causes gravity to move it down over hair cells. So these hair cells will synapse with neurons and fire very simply from the mechanical change through the vestibular branch of the vestibulocochlear nerve. Now, we have fluid in the inner ear. Very important for equilibrium. Does anybody know why? We look at very simply carrying a bowl of water to help us explain how this works. So very simply, and all of us have done this, if you carry a bowl full of water, what happens when we have a very sudden stop, a very quick start? What happens to the water in particular? It usually sloshes because the water is constantly in motion. And also there's a little bit of a lag time, so as you move, the bowl of water doesn't exactly move with you. It's going to take a split second for it to catch up to that particular type of motion. And the second thing to think of too is what happens when you rotate very suddenly. Same thing. Some of the water is actually going to slosh out of the bowl very quickly. Some is going to go with you, and then as it catches up and you stop, the water is going to keep going. It's that quick little change of position or that lag time that we find a fluid in the ear that helps us detect positions in equilibrium, both from a static position as well as from a motion perspective. That's what the otoliths and also the crystals are basically there for. It's that little fluid gelatinous motion that gives that split second or lag time that gives us the detection of what kind of motion we're basically experiencing. So here's a quick look at the section of the macula. You're going to find the otolithic membrane and the otoliths, and these are all your crystals into here. So basically they're going to embed the hair bundles of the hair cells, and these are going to be known as stereocilia. So again, you have a sensory and a motor fiber. The motor fibers are there to help maintain tension along those particular areas. You're going to find these deep within the utricle and the saccule. So again, utricle for linear or horizontal acceleration or deceleration, saccule for vertical acceleration and deceleration. And one strange thing that happens, and we'll get into this when we talk about aging in particular, the otoliths and otolithic membrane is somewhat fluid through the majority of our mature life. As we get older, unfortunately, some portion of the population, the otolithic membrane starts to harden. So what ends up happening is we can't detect quick sudden changes of motion. This can lead to issues such as motion sickness, dizziness, vertigo, and so on and so forth. And unfortunately, as far as I know anyway, there's no real fix for this once the otolithic membrane starts to harden. As far as I know, I'm not sure exactly what the exact mechanism is of why this particular thing happens. Show that certain people do experience this. So you have a close-up shot of the hair cells. You see the otoliths, or the crystals, sitting on top of the otolithic membrane. And you see the hair bundles of the kinesiolium. That's basically the opening where the hair bundle will list. And you see the stereocilia there. This is going to contain the gated channels, the potassium gated channels, that will, once we see any kind of vibration with the otolithic membrane, it'll mechanically open up the potassium channel cells, which will basically trigger the hair cells to release a neurotransmitter, and basically has a synapse there, as you can see, with the vestibular cochlear nerve, and fire off the action potential. Now, the three semicircular canals are very responsible for the dynamic equilibrium, so these are going to be very important for rotational acceleration or deceleration. So they have very similar format, but it's a little bit different in some aspects. They have an ampulla, which is going to contain crista, and also has its own group of hair cells. So very simply the same thing. So its movement of the head will very simply have a lag time with the ampulla, which affects the endolymph of the hair cells. So this will also bend the hair cells and fire off the action potential. So again, this also synapse with the vestibular branch of the vestibular cochlear nerve. So here we see the ampulla found deep within the semicircular ducts. So you see the actual cupula, which is actually the portion of the otolithic membrane here, with otoliths found therein. You see the same situation here with the crista, the hair bundles, and also the hair cells. So again, you have different portions of the sensory motor fiber that's going to attach to the ampullary nerve, and you'll see in a couple seconds here what kind of changes with rotational motion and how it's detected. So here we see on the left, we see a neutral head position, so the person is facing forward. The cupula and ampulla are lined up perfectly. So in this case, the hair cells are in a resting state. So in this case, when the person starts to rotate, now in this case, looking at the person, they're going from the head from the rotated position to the left and rotating back to center. So what happens here, the cupula gets dragged in a certain position here. So that little bit of a lag time allows the crista to allow the hair cells to bend, which basically allows the potassium to rush in and fire off the action potential. And this gives us the detection of angular motion, in this case, rotation. And this follows a slightly different pathway than we saw for hearing. So again, from the vestibular portion of the cranial nerve 8, it's going to synapse in the vestibular nuclei, also in the spinal cord, down the vestibular spinal tract. It's going to synapse with the cerebellum, as well as the abducens nerve, the motor nucleus, the trochlear nerve, ocular motor nerve as well. These all feed into eye motion in particular to help kind of reset our eyes over the horizon. And finally, it'll synapse at the ventral posterior nucleus of the thalamus, as goes to the vestibular area of the cerebral cortex. Notice it's just superior to the cochlear area of the cerebral cortex as well. Now, because it synapses with those three nerves, the three current nerves you see there, and that should trigger something in you, 3, 4, and 6. I'm sorry, 3, 4, and also that should say 6, not 5, under abducens nerve. These are all nerves that control eye motion. So very simply, these are very tied into keeping your eyes level with the horizon. So very simply, when you guys are doing the Special Senses lab in particular, and unfortunately you won't be able to use the balance boards or Bosu balls, I would have brought some into the lab to do so. These are all ways to help maintain your posture, your brain and your the inner ear, as well as your cerebellum, and also the portion of the midbrain and the brain stem will help bring your eyes over the horizon as best possible. So if you were to do any kind of balancing exercise, your eyes help maintain position, so does your inner ear, so does the cerebellum, and also the proprioceptors found all throughout the joints, all throughout the body. Now, if you close your eyes, however, you lose one sensory input as far as trying to maintain proper position. So then you're left with your cerebellum and also left with the inner ear to help maintain position. This, for some people, can be very disorienting. So now you're relying upon, this is the answer to one of the questions for Special Lab, you're relying upon your cerebellum, you're relying upon the inner ear, the vestibular branch, as well as all the proprioceptors found within the joints to help maintain your body positioning in space. Now, this can be thrown off even more so those of you who are ever gone swimming in deep portions of a pool or deep portions of the ocean, or have gone scuba diving. This, for the most part, can keep you somewhat upright. But because you're free floating, for the most part, you can get very disoriented very easily. It does throw off the inner ear quite a bit. Also, you find this with astronauts at the same time, trying to find what the quote-unquote down position would be. This really messes with the inner ear in particular. So again, taking away the visual input of maintaining your eyes level with the horizon can severely mess with your equilibrium. But that's something that can be improved with balance exercises, which, again, if you were to do a test of special senses with a boosted ball or a balance board, that's something we use for rehabilitation and also for rehabilitation of people that have vertigo, dizziness, and other equilibrium or balance issues. So the next couple of tables here gives a quick synopsis of the major structures and a very brief overview of each function of each particular structure here. So here we see the outer ear with the argol, the external auditory canal and tympanic membrane, middle ear with the auditory ossicles and the eustachian tube, and the inner ear, of course, with the cochlea and the vestibular apparatus. We'll go very briefly through the embryological development of the eyes and ears. Very simply, the iris will develop at 22 days. The ectoderm of the forebrain forms the optic grooves. And to be quite honest, I probably will not ask too many questions based on this. Just very simply that the eyes are going to start developing about 22 days after fertilization. The rest I'm really not going to test you on. And you can skip this as well. And skip this. Yeah, very simply, if you also notice, the internal ears also start to develop around 22 days after fertilization. And skip this as well. Okay, so as far as aging and special senses go, so obviously some of these are going to be very self-explanatory. So smell and taste usually does not cause much of an issue until you get past the age of 50, and of course that's a very round number. Some people it's lost more quickly. Some they maintain their smell and taste well into their 60s or 70s. But for smell and taste in general, we start to lose receptors, or very simply the regeneration just takes a longer time to regenerate certain portions of the olfactory and gustatory receptors. So a lot of times long term we see happening is they can't smell any kind of obnoxious smells. A lot of times food tastes very bland. That's why a lot of times older people may like things a little more spicy, with a little more different types of flavors to it. For the most part, smell and taste does diminish quite a bit after 50. Now, as far as the lenses go, this is debatable. Around the age of 40, you start to see loss of elasticity of the lens, and then of course you start to have problems with nearsightedness. You do see high prevalence though of 40 to 45 50-year-olds. Very simply, they need bifocals or at least get transitional lenses, but they do lose the ability, even with glasses, with normal glasses, to see things that are very near to them. So reading menus, reading books, you work on the computer, they tend to have major problems with that nearsightedness. Naturally, the irises also start to weaken and react more slowly. That's why a lot of times you'll see elderly people will wear, the very elderly will wear the special sunglasses that encompasses the entire vision, because they have very much difficulty adjusting to changes in lighting. Usually worse with trying to detect with detection of light, and of course with dark adaptation, it takes a longer time than normal to adjust to the dark. And naturally couple that with problems with joints or weakness in muscles, naturally these people can fall very easily in dark environments. And of course we see different types of diseases as well. We see retinal diseases like macular disease, detached retina, and glaucoma. Unfortunately, these happen very frequently in the elderly. Some medications can help with some of these, but most oftentimes not. Surgery is usually mandated. Usually by about age 60, again, they start to lose some of their hair cells, which is known as proboscis. About 25% of the individuals will notice hearing loss. Very simply, what happens is certain hair cells will vibrate and die. That's why a lot of times they'll talk about a ringing in the ears that lasts for a couple seconds and all of a sudden just stops. Now, in the more severe cases, we hear patients that will have tinnitus, known as ringing in the ears, and this can range to a couple decibels to a very loud, almost to the roar of a jet engine. Most oftentimes, this can be something that's idiopathic, where very simply it's something that just kind of comes on. Some people, if they're in certain environments, like they are in certain industrial environments, or they've been in certain wars, or again, they dealt with airlines, dealt with airplanes, or so on and so forth, or even musicians that have been dealing with a lot of high decibel sound waves with damage to the ears can experience tinnitus later on in life. And unfortunately, tinnitus, unless you take different types of pain medication, there's not much you can do for it. And finally, we have vestibular imbalance, which comes again as no surprise. So a lot of the elderly start to lose their balance because their brain simply cannot perceive positioning or be able to basically provide feedback as they start to walk. And then again, couple that with muscle weakness, joint abnormalities, arthritis, and so on and so forth. You can imagine the challenges to the vestibular area trying to maintain proper balance. So this ends chapter 17. This is the completion of the semester as far as the narrative material goes. So again, the final exam is going to encompass chapters 13, 14, 15, 16, and 17. A little smattering of some of the earlier chapters, but for the most part, it's already covered within these chapters we just mentioned here. That's it. If you guys have any questions, feel free to email me. Most of the special senses labs can be answered through this lab, through this narrated lecture, as well as the lecture that deals with the sensory, integrated, and motor systems. Those two narrated PowerPoints really should be able to help you answer the questions for the last lab. All right, thanks for hanging with me, guys. I know it's been quite boring listening to me drone on, and I'll talk to you guys soon.